Okay, so I'm Misha and I'm going to be talking about acute inflammation. So first of all, when you talk about inflammation, inflammation is basically a body's response to any kind of damage. At basically, we are attempting to maintain homeostasis. Now, it is basically made up of two parts, but here, first of all, let's talk about the stages. Now, acute inflammation can be divided into morphological phases and then stages according to what's basically happening. Morphological phases is what you would see, like alterations, exudation, proliferation, which I'll be explaining later. And then the processes that are occurring. So there are five R's, which is recognition, recruitment, removal, regulation, and resolution. So these two things are absolutely very, very important that you need to mention when you're talking about acute inflammation. Another thing is you already know that patho is basically about what you can see macroscopically. So they'll ask you about the cardinal signs for sure. So you need to know these five things. You need to know redness, heat, swelling, pain, and dysfunction. Or you can remember it as the other way, rubor, tumor, calor, daller. And basically you can see the reasons why they're occurring as well. Now, some professors may ask you about like what are the mediators that are causing it, but that's mainly pathophysio, but you can know it if you want to. Now, acute inflammation can basically be divided into two parts. Either it can be vascular or it can be cellular. Vascular, the events that are occurring on the vessels, obviously. Cellular events are we know that previously you've already learned that acute inflammation is basically neutrophils, right? And chronic inflammation is macrophages. So cellular events would be when you're talking about how they're migrating, how they're moving into the tissues. Vascular events is what helps them do that. Okay, so another important thing that you need to understand is that when we're talking about acute inflammation, the time that they're occurring or for how long they're occurring also varies. So normally acute is less than 2 weeks. I talked about morphological phases. So there are three morphological phases: alteration, exudation. Basically, it's the same thing as those five R's, but more of what you would see. So, alteration, attack by agent, exudation, fluids, pus, our body basically reacting to that particular attack. And proliferation is either we would have regeneration or repair, which you'll be learning about. These are two very different things. And basically, you'll be learning about regeneration and repair later on. Now, obviously, there are different outcomes. Our body not always, cures with acute inflammation, or there's not always repair or regeneration. Sometimes you go into chronic inflammation, sometimes acute inflammation keeps going on. It depends on what is the situation. And if we talk about the cell and fluid domination, so throughout the acute inflammation, first of all, immediately we would have fluid exudation. After 24 hours, neutrophils will start to move in or they'll peak. And then 2 to 3 days later, we would have our phagocytes come in. And that's like within like 2 weeks, we would have either the transition into chronic inflammation or acute inflammation would just keep going on. Now, here comes the main part. So, when we're talking about in acute inflammation, and these five R's are very important because this is what is happening. So, first of all, there's recognition. Recognition is basically here we talk about TLRs on WBCs. So, toll-like receptors, which are present on the WBCs, will actually interact with the PAMPs or the DAMPs, okay, which are pathogen associated molecular proteins. And once the interaction happens, this leads to the activation of tissue factor called NF-kappa B, which causes production of other cytokines like TNF alpha, interleukins. And again, as I mentioned, that mediators, if you learn them, easier for pathophysio. If you don't need them for patho. Now, recruitment, and the second stage is recruitment. Before we go into recruitment, so these are basically all the phases. And in recruitment, one very important thing that you need to learn is that we are talking about vascular changes over here. Okay, even though there are also cellular changes, first let's talk about the vascular changes. Either First of all, we'll have vasodilation, relaxation of the arterioles. Now, this can be due to histamine, bradykinin, prostaglandins, nitrox oxide, and increased permeation. Now, this is very important. By contraction of the endothelium in the post capillary venules. So, you need to remember where they occur because they will ask you about where this increased permeability is occurring. And this is also mediated by histamines, leukotrienes, prostaglandins, and bradykinins. The permeation basically allows albumin to escape, and if albumin escapes, what happens? We have edema, right? And that's the tumor that they say previously, because whenever they saw swelling they thought it was tumor, so that's the reason even edema is in Latin is still called tumor. Now, there are obviously changes in the lymphatics as well. The increased edema means more lymphatic flow. If the micro debris or WBCs enter the lymphatics, we would have lymphangitis. If they reach the nodes, we call it lymphadenitis, which may be painful. And then we can have reactive lymphadenitis, which is hyperplasia or reactive hypercellularity due to proliferation or germinal centers in supporting of inflammation. So basically, again, very easy. If there is recruitment of these WBCs or debris or microbes into the lymphatics, it will cause lymphangitis. Okay. Now, another thing is that we talk about four steps which are leading to extravasation. So here we can see how are these neutrophils actually entering into the tissues? How are the leukocytes entering into the tissues? First of all, we have margination. Margination is that we are having vasodilation, which causes neutrophils to flow along the wall instead of the center of the lumen, as we can see in the diagram. Then we have rolling, which is histamine, tissue necrosis factor alpha, interleukin 1 causes expression of selectins on the endothelium, as we can see P selectins over here, P selectins on the endothelium, which interacts with sialyl Louise X on WBCs. Okay. So we can see that there's an interaction over here. Once they're bound, adhesion occurs. TNF alpha interleukin 1 causes expression of CAMs on the endothelial cells. Here we can see integrin ligands, right? So CAMs on the endothelium, which interacts with the integrins on the WBCs. Now, the reason that I'm telling you all of this or why you need to know is because there is pathology related to it. So, there are diseases such as leukocyte adhesion deficiency, where the integrin is faulty, right? So, the interaction does not occur, and so the migration, or we can say, yeah, the migration basically cannot occur. The next step, the fourth step that we can see over here is transmigration or chemotaxis. So neutrophils will squeeze or we say diapedesis through post capillary venules. Always remember all of these processes are occurring within the post capillary venules, not in the capillaries themselves. So they move into the interstitium. Now, here again, we have diseases, and that's the reason we need to know this. We have lazy leukocyte syndrome, where WBCs don't migrate from the bone marrow, and we have chemoattractants when outside. When outside of the vessels, C5a, LT4b, interleukin 8 show the way, often produced by tissue macrophages and other cells. Next, we can talk about removal. So, one second. Again, here I've highlighted that it's post capillary venules. Anyways, so we can go to removal. Now, removal is the invader must be phagocytosed or it should be destroyed. Now, phagocytosis is through pseudopods will extend, a phagosome forms, fuses with the lysosome to make phagolysosomes. This attraction is random unless marked by opsonins, right? So in this case, again, the reason that I'm mentioning this, it's a very easy concept, but there's a disease which is Chidiac-Higashi syndrome, where there's an inability to actually form the phagolysosome. Again, then the removal cannot occur. Our whole process of this doing this is that we can remove the agent which is causing the particular damage to the tissues. Then secondly, we talk about destruction. I said that neutrophils are the main cells that are involved in acute inflammation, right? So destruction can be either oxygen dependent or it can be enzyme dependent. Oxygen dependent is by the neutrophils. They use HClO, which is bleach, from myeloperoxidase, whereas enzyme dependent is by using lysozyme and other enzymes. Now, there can be some diseases where we say disease of oxygen dependent killing, which is CGD, where there's bad NADPH oxidase, so no superoxide is present. In this case, the neutrophils do not actually have the ability to produce H2O2. So what they do is that they use the bacteria's H2O2. But if the bacteria is catalase positive, then it won't generate that, and then it would get very difficult to actually get rid of that particular bacteria, for example. And finally, we go on to regulation. Now, obviously, if acute inflammation is not checked upon, for example, if it keeps going on and there is, uh, there is no stop to it, right? What happens is that inflammation is good for us, but if it's going on for a longer period of time, eventually it starts to damage our own tissues as well, right? So we say that the process can proceed to harm the rest of the organism. So, how do we prevent this? So, again, here comes the mediators. We have anti inflammatory mediators and self limitations. So, we have no more agents, no more cytokines, cessation of the vascular effects, and no more chemotaxis. So, neutrophils will die within 24 hours, and we have stop signals that are produced by macrophages like interleukin 10, TGF beta are anti-inflammatory agents. And finally, we can go on to resolution. Now, resolution best best outcome would be healing, right? So resolution and healing. The vessel permeability it decreases, and constriction also returns to normal. The migration will stop, and tissue healing begins. Either regeneration or repair. Again, I said that you will be thought about that in the later videos. And or so this is the best one that can come out, or and we can have other outcomes depending on the situation. So we can have a continuation of the acute inflammation if the macrophages are secreting interleukin 8, which keep recruiting neutrophils. So we have more pus, right? As I said, we have exudation. So exudation, we can have pus, for example. Then we have progression of, or it can progress into chronic inflammation. Now, this does not always occur. There are some diseases in which we go directly into chronic inflammation and don't have acute inflammation. Or we can have acute inflammation which goes to chronic inflammation, or we can have only acute inflammation. So in this case, we're saying that there's a progression to chronic inflammation. So macrophages present MHC class 2 to CD4 cells, and we know the human immunological pathway that occurs, and that is basically our chronic inflammation. And then we have abscess formation, which is containment by creating a fibrotic shell. Again, it is important to remember that I said a fibrotic shell, they do not have an epithelial lining. And what this shell does and why we have the formation of this cell or this abscess is basically around the invader so that it doesn't actually spread. So our body just like it contains it in a particular location. So we can prevent it from spreading. Or we would have the worst case scenario is that they would be death of organism. So basically this is acute inflammation. And I think if you go through the presentation, you can see that there's the notes that I've written on the side are actually the most important things that they would ask you if you get this question. But and about the mediators, you can know them because they'll help you eventually for pathophys. But it's better to just know them in case they ask you. So yeah, that's all from me. Thank you.
Right.